Hello, welcome to my talk. I'm Chris Whips. I'm from the State University of New York, College of Environmental Science and Forestry, or just known as SUNY ESF, and that's in Syracuse, New York. So today my talk is on mycobacteriosis in zebrafish, with an overview of some of our findings over the years, as well as some recent findings on line susceptibility in zebrafish. First, I'd like to acknowledge the many great collaborators and long-term funding sources I've had over the years. Funding has been primarily through an NIH grant where Mike Kent is the lead PI on that. I've been a collaborator with him over many years, including I did my PhD with Mike Kent, so we've had some great years of collaboration. Also, many folks in my lab over the years, and particularly the stuff I'll be presenting on today, comes out of work from Carolyn Chang, who was a PhD student of mine, and a master's student, Andrew Janik, with some of the more recent findings. So I like to show this slide. I made this several years ago. This is a summary of some of the data that's freely available from the Zebrafish International Resource Center and their health diagnostic services. And I just want to point out here, we've got sort of the number of facilities positive for certain pathogens over this 10-year span. This Pseudoloma microsporidium parasite comes up at the top, and then Mycobacteriosis, the general disease, that comes up next. And I always contend that if we really went looking, we would probably find a lot more mycobacteria in zebrafish systems. These are just common bacteria that you will find in aquatic systems and in fish in general. So why do we care about any of these health issues? Well, the obvious one would be, well, if your fish are dying, you can't do your work. Maybe more of a concern even would be the morbidity, this idea that pathogens can act as a source of variation in experiments that you may not be aware of. So you're carrying out your experiment, you don't know your fish are infected with something. It's not so bad that it's killing the fish, but it's impacting the results of your experiment in some way that you are not aware of. So that's a concern. And the third thing on the list here is at least one species of Mycobacterium, Mycobacterium marinum, is known to cause skin infections in people. This has been known for a long time, and then in recent years there have been reported cases of folks in zebrafish facilities acquiring these skin infections. So mycobacteria in general, you might be familiar with because Mycobacterium tuberculosis and Mycobacterium leprae are important human pathogens. But these are common pathogens in other animal systems as well. There are over 190 species in the genus. The kinds of infections, so Mycobacteriosis, the disease, it can range from acute to chronic disease, so more quick-acting to these long-term smoldering type infections. They're difficult to treat, whichever ones we're talking about, humans or other animals. Difficult to treat from an antibiotic perspective. And then just working with them is challenging. Some of these are difficult to grow on, say, a culture plate. Some of them take weeks to grow on a culture plate, so not as easy as some other bacterial systems. And then what's really important with our zebrafish ones is, one, mycobacteria can be environmentally derived, and two, they can form surface biofilms. So in an aquatic system, mycobacteria will happily live on those surfaces whether or not, say, fish are present, so they can persist in the environment without the host being present. And that's illustrated to some extent here. If we look at surface biofilms, this is from my own data, collecting surface biofilms with a swab from a tank from many different facilities over many years, we find this long list of different mycobacteria. If we look in fish, we don't find as many. So there are some species of mycobacteria we find on surface biofilms, but we would have never encountered in fish. So there's some kind of some reason some of these can be pathogens in fish or not. And that's something we're trying to understand. And then, of course, just a reminder here, we do have the marinum being a potential cause of these skin infections in people. So I'm really trying to understand this is, how do we go from something that lives in a biofilm to something getting in zebrafish? And then when it's there, which of these are sort of the worst for zebrafish, and how can we deal with them? So one of our challenges working with adult fish is that you can't really determine the infection status of an individual live fish. You typically need to do your experiment and then see later. So we would determine whether a fish is infected by histology or PCR or culture. And that creates kind of a problem because we always, you do your best to set up the experiment well, and we've done this for many years, but you can't tell how the experiment's going in the middle of it unless you, of course, have mortalities. That might be an indicator of infection, or you take some fish out as a sample along the way. So what we've been doing more recently is using Casper fish, where we can see through the body wall of the fish, and we use a fluorescent Mycobacterium colony, and therefore we can monitor these infections over time. So here's an infected fish. This is a cartoon, obviously. Imagine this is a Casper fish. And when we look under fluorescence, we can see right inside the fish, and we see these glowing dots. These are the granulomas, these aggregates of mycobacteria and host cells that tell us that this individual fish is infected. So I'll give you some examples of how we've applied this system, as well as using Casper fish and other zebrafish lines to look for differences between them. So first we'll start with the biofilm example. This is work we published several years ago, but I think it bears repeating, and it shows how we can apply this fluorescence and Casper fish system pretty easily. So remember, mycobacteria can live in surface biofilms, and one of our big questions was, how soon can we detect mycobacteria in biofilms or detritus in a tank if we have infected fish? And then the other question we want to know is, well, if you have mycobacteria living in biofilms, how soon do fish get infected? How soon can that occur? And so we set up two different experiments, and this work was done by my PhD student, Carolyn Chang. Lots of help here from Jet Lewis, who was one of our undergrad research assistants. And the first experiment was this: infect fish, either by injection or through feeding of an infected food that contains these fluorescent mycobacteria, and then put the fish in a tank and monitor those biofilms and sample the feces and detritus weekly. So we're looking to see when can we start to culture and detect mycobacteria in those surface biofilms. And I mentioned this has implications for monitoring, right? Because if you want to understand, do you have mycobacteria in a system, you could take detritus from a tank and look for the mycobacteria. How soon would you find that out? We'll get to that. The other experiment, though, first is, you've got bacteria in the biofilms. When do you start to see it in fish? So what we did, we grew mycobacteria with the fluorescent mycobacterium on the surfaces of tanks, just with water, no fish in them at this point. We allow that biofilm to establish, and then we would add fish to those. And then using the Casper fish that are individually tagged, we use elastomer tags, and we can tell individuals apart, is see what happens over time. So we look every 2 weeks, and what we saw was an increasing prevalence of infection in those fish over time. And so this is work that's was published in 2019. If you want more details on that, you can check that out. Bottom line is this. When you have infected fish, you start detecting mycobacteria in those biofilms within a couple of weeks, so pretty quickly. So that means that this is probably pretty good for monitoring if you've got an infected fish. The other way around, within 2 to 4 weeks, fish start getting infected, or I should clarify, we can detect it in fish. Fish might be getting infected earlier than that. And likewise, with the biofilms being seeded here, this is when we start to detect it. This has a couple of implications. One is, if your tanks get really biofilmy, there's probably mycobacteria in there that are acting as a source of infection for your fish. And then, of course, with the other way here is you can use the biofilms to monitor for mycobacteria. It's really important, though, to remember mycobacteria just writ large, they're ubiquitous in aquatic systems. So if you're looking at surface biofilms or detritus, you really want to target those specific mycobacteria that are problems that could have come from the fish. If you just say, let's see if there's mycobacteria, you might find them, and that doesn't necessarily tell you that they're in your fish. All right, so how about dealing with mycobacteriosis through treatment? So we've tried this. This is work that we published a few years ago looking at tigecycline and clarithromycin over either 2 weeks or about 4 weeks in terms of treatment. And I'll just jump right to the conclusion of this particular experiment. This is work from Carolyn Chang's thesis again. We didn't see a reduction in prevalence of mycobacteriosis in our fish, but we did see reduced severity in treated fish. So these groups with the lower bars, these are all our treated fish. So this seemed promising to us, and we decided, let's take this further and apply our Casper and fluorescent mycobacteria model with the idea that we could treat the fish for longer periods of time and not have to worry about when the end of the experiment is, like when do we decide to end the experiment, because we can look at the fish and see what happens over time. Hopefully a reduction in these, little spots of fluorescence that indicate an infection over time. And so this is work from Drew Janik's thesis. Again, and what we did, we tried higher doses of antibiotics, so a few different doses. But again, tigecycline and clarithromycin like we tried before. And this is through feeding, so a gelatin feed that contains the antibiotics. And I'll just jump to the conclusion, really, because it's actually pretty similar to the conclusion that we found before, which is we did not see a reduction in prevalence like we had seen before, even with higher doses of antibiotics. And this figure up here shows a lot of detail here, but you can just get a sense maybe from looking at this no antibiotics. So those are our infected fish. And you can see the bars, no infected fish and no antibiotics. And the bars are pretty similar to those from fish that were treated with clarithromycin or tigecycline. So we didn't see an obvious decrease in that green fluorescent protein over time like we were hoping. We did not see a reduction in prevalence, but like the earlier experiment, we do see a reduction in these granulomas whether or not they contain those acid-fast bacteria. Those are mycobacteria. So we do see that helping. So, what do we take from that? Well, sort of the same thing as we learned before. Antibiotics can reduce the severity of infection. That could mean they're useful for cleaning up valuable stocks of fish. The doses that we used in the Chang study a few years ago at the lower doses, those are probably good enough. The higher doses didn't seem to make a big difference in our more recent work. And we know these lower doses are well tolerated in terms of the fish, say, being able to breed. So we definitely do not recommend using antibiotics just broadly in a system if you have mycobacteriosis, but only in maybe a targeted way where you're trying to lower the severity of infection in a valuable line of fish so you can breed them and disinfect those eggs and hopefully get rid of, escape the infection in that next generation. All right, so the last thing I'll talk about today is mycobacteriosis in different zebrafish lines. We know that different mycobacteria can cause different manifestations of disease, and we also know from some earlier data that it seems that maybe some zebrafish lines are more susceptible to infection than others, or have higher prevalence in general for whatever reason. And the early data came from some survey work that I had done many years ago at a facility looking at different zebrafish lines, and we consistently found that TU fish had a higher prevalence than the other fish we were looking at. This was actually followed up by Katie Murray a few years later. She also found similar results in that TU fish seemed to have higher prevalence than other lines of zebrafish. So what we wanted to do is, using our fluorescent Mycobacterium and Mycobacterium coloni and Casper fish, as well as these other lines of zebrafish, is see in an experimental setup, do we see differences in prevalence and severity? The work was all done by injection exposure. Put a pin in that. That will be important when we think about what might be going on with some of the differences here. And this is work that's just recently published and came out of the master's thesis of my former grad student here, Drew Janik. All right, so getting right to the results. So we infected fish, let them sit for many weeks, and then look and see what do we find. We looked at green fluorescent protein, so these are in dissected fish. We can't see through the body walls of these other lines of fish like we can with the Casper, so this is a terminal point of the experiment. And what we found, say just looking at GFP, was pretty similar prevalence in all the groups. The other thing we found, unfortunately, was some infections in our control fish, which were TU fish as well. These were meant to be uninfected. We know from our starting fish were likely uninfected, and so we believe that this contamination occurred sometime during the experiment. And we still believe that we can make comparisons between these different lines of fish nonetheless. We also looked at histology, so tissue sections, and then image analysis, which is a way of kind of taking some of our subjectivity out of the visual evaluation of the severity of infection. And in all these cases, I'll come back to histology, in all these cases, the prevalence is very similar in all the groups. Where we did see differences, kind of like with the antibiotic experiments, is in severity. So prevalence, okay, looks pretty similar, but severity was different. Severity was much higher in TU fish. And again, unfortunately, our controls as well, these were also TU fish. But that compared to 5Ds or ABs or Casper fish, there are bars here, they're very low for the Casper fish. Severity was pretty low. So this still supports the idea that TU fish are more susceptible to these infections, or the infections can become more severe in TU fish. Another interesting finding was with the 5D zebrafish. Although looking at granuloma counts, they were pretty low, we did see very disseminated infections in these 5D fish. We see these glowing spots everywhere. So the mycobacteria really spread through these 5D fish, even though they were less sort of focal, forming these granulomas like we see in these other fish. So that's interesting. Again, suggesting there's genetic differences between these fish, and that may be driving the differences that we're seeing in the manifestation of these infections. All right, so finish up. A reminder back to that idea that we injected all these fish. So we're really circumventing any sort of natural exposure mechanisms or resistance mechanisms from the fish perspective. We still did see differences in severity of disease with the TU fish in particular, but not prevalence. So it would be interesting to do this again, but using a more natural route of exposure. So TU fish definitely seem to be more impacted. The fact that 5Ds had a very different look to their infection, much more disseminated, again, suggests there may be a genetic basis to this. And doing this experiment again, obviously we would really want our control fish to have no infection at all. So plenty of work still to do, and we're working on these more natural exposure models and looking forward to finding out where we go from there. All right, thank you.